Okay, so I'm a little late getting your Chapter 6 lecture done. So I'm going to do the Chapter 6 and Chapter 7 lectures together because both of those PowerPoints are in this one PowerPoint, so we can look at that. Okay, so when we did Chapter 2 on macromolecules, we talked briefly about DNA. At that time, we talked about, we talked briefly about the structure of DNA. We talked about DNA is a nucleic acid molecule. It's a long polymer. It's formed by the subunits called nucleotides. It's formed by covalent and hydrogen bonds. And so if you want to review some of that basic structure, you can go back and look at Chapter 2. We're going to look at a little bit more detail about the structure of DNA in this chapter, and we're going to talk about some of the things that DNA does. DNA can do two things, and so we're going to talk about both of those things in each of these chapters, in Chapter 6 and Chapter 7. The first thing that it can do is it can make copies of itself, and that's called replication, DNA replication. And the second thing that it can do is it can be used as instructions to make proteins. And the steps of making proteins are transcription and translation. So Chapter 6 does the DNA structure and the DNA replication piece. And then Chapter 7 talks about the transcription and translation piece. So Chapter 6 starts by looking at how DNA is in the cell. So in the previous chapters, we talked about the cell structure. We talked about the organelles that make up the cell structure. We talked a little bit about cell function as far as enzymes helping to do chemical reactions. And so this is trying to correlate some of that information with cell structure with what we're getting ready to talk about DNA and what DNA does and making proteins. And so we talked about two different types of cells, prokaryotes and eukaryotes. Since we're talking about the nucleus and other types of organelles, we're talking about eukaryotes here. And in eukaryotes, we organize the DNA into chromosomes. So a eukaryotic chromosome is a molecule of DNA, and we're organizing that DNA by wrapping it around proteins. And so that chromosome, which is DNA wrapped around proteins, has sections of DNA. And those sections of DNA are called genes. And that gene is the instructions for making a protein. And so when they first discovered this, they came up with what they called the one gene, one protein hypothesis. Or the one hypothesis. That's a good spelling of hypothesis, isn't it? And so that one gene, one protein hypothesis is the fact that a gene leads to the production of a protein. Well, now we know that's not always the case. That's not always true. We can have a gene that can make a couple of different protein products. But the thing that is true is that proteins come from genes. The genes contain the instructions for making proteins. There are multiple genes on each chromosome because the chromosome is a long strand of DNA that's wrapped around proteins. In this image from your textbook kind of shows that. It shows the DNA double helix. That's the structure of the DNA that we talked about a little bit in Chapter 2. It shows them wound around these proteins and then all of the DNA around the proteins together associate into these larger and larger coils that make these chromosomes. And then all the chromosomes line up together in the cell in structure, in situations like when we have mitosis. This image that's shown on the left is a scanning electron micrograph of an actual chromosome. And it looks fuzzy because what it is the increasing levels of organization of these DNA strands. These strands of DNA wrapped in a double helix, wrapped around these proteins, and coiled up to form these chromosomes. This slide gives you some genes that we use to associate with the chromosome structure. And this also shows what happens when we duplicate chromosomes. So for part of the cell cycle, for part of mitosis, we have a single chromosome that's unduplicated. And then right before the cell divides, right before the cell undergoes mitosis, that chromosome duplicates itself. And the duplicated part is sometimes called a sister chromatid. And they are attached together at an area called the centromere. So the eukaryote has DNA that's organized into these chromosomes, and each individual species has a characteristic number of chromosomes. And so that's one of the reasons why in the discussion board what we're doing is we're looking at different species. We're figuring out how many chromosomes they have. So the chromosome number is all the chromosomes that they have. Most chromosomes in body cells in humans and vertebrates occur in pairs, homologous pairs. Homologous means similar. Homo means same. Homologous pairs. So a human body cell has 23 pairs of chromosomes, or 46 chromosomes in each cell. So 46 is the chromosome number for humans. And because they're in pairs, when we talk about that 46 number, we call that the diploid or the 2N chromosome number. So cells that have that 46 chromosome number, body cells in humans, are diploid or diploid cells. And then cells that only have one copy of each chromosome, a lot of times these are sex cells or gametes. In humans, these are egg and sperm. We represent the number of chromosomes as N, and we call these haploid cells because they only have one copy of each chromosome instead of two in the diploid or diploid organisms. And so we have some examples of that, some examples of chromosome numbers. Fruit fly has 8 chromosomes. Potatoes have 48. There's some that have quite a lot of chromosomes. There are 2 types of chromosomes in eukaryotes. The majority of our chromosomes are autosomes. These are paired chromosomes. Humans have 22 pairs of these autosomes. You get one from your mother and one from your father, and that's how you get a pair. One came from the egg and one came from the sperm that formed you, and they've paired up, and they have formed these 22 pairs. And then the last pair, the 23rd pair, are what we call sex chromosomes. This is a typo. Humans have one pair of sex chromosomes. That's why it doesn't have an S on the end. Humans have one pair of sex chromosomes. These differ between males and females, and these actually determine sex or gender. In humans, we call them the X chromosome and the Y chromosome. If you have two X chromosomes, we declare you to be female. And if you have an X chromosome and a Y chromosome in humans, we declare you to be male. Sorry about that typo. I'll take a note, change it. In PowerPoint. And so that's what this figure is showing you. The figure starts by showing you the autosomes, the normal body cells. They're going to have these as a pair. So these are diploid cells. And they go through a process called meiosis. This is different from mitosis. This is the way that we make egg and sperm cells. A female will make egg gametes, and they will have an X chromosome in them, either this X chromosome or this X chromosome. Either way, it's an X chromosome. The male can make two different kinds of sperm. The male can make a sperm that has an X chromosome or a sperm that has a Y chromosome. And they're showing you a Punnett square, which is a way of representing genetic crosses. We're going to get more into Punnett squares later in the semester. But if the X from the male and one of the X from the female come together, your offspring is an XX. If the X from the male comes together with the other X from the other female, then your offspring is also female. If the Y from the male comes together with one of the X's, your offspring is XY, which will be a male. If the Y comes together with the other X, again, XY determines the male. So it's these gametes, or these sex cells, that determine sex of the offspring. That's his offspring, even though it went off the side. So what they're showing you, or what they're representing to you in these cells, are sex chromosomes, X and Y chromosomes. And those chromosomes are long strands of DNA. So we can look a little more at that DNA structure. DNA molecule is two strands of nucleotides that are held together. They run in opposite directions, and they're coiled into a double helix. The nucleotide is the basic subunit of DNA. So a DNA nucleotide is made up of a sugar and some phosphate. And the important part of the DNA nucleotide is the base. There are 4 bases in DNA: adenine, thymine, guanine, and cytosine, and we abbreviate those based on their first letter. Adenine we abbreviate with an A, thymine we abbreviate with a T, guanine we abbreviate with a G, and cytosine we abbreviate with a C. The 2 double helix strands are held together by hydrogen bonds, and so anything that disrupts hydrogen bonds can disrupt this double helix structure. And then we have some base pairing rules that we talk about in regards to the DNA. These are also called Chargaff's rules. A and T pair together, and C and G pair together. So Figure 6.5 shows you the structure of each of the nucleotides. The part that varies is this base. You don't have to memorize any structures. But the adenine and the thymine pair together, and the guanine and the cytosine pair together. This is a large one pairing with a small one, a large one pairing with a small one. And once they base pair together and form hydrogen bonds with each other, the A and T can have 2 hydrogen bonds that hold them together. The C and G form 3 hydrogen bonds that hold them together. Then this whole thing forms this double helix structure. And that double helix structure is what we think about as the DNA, and that's what I showed you in that previous figure that coils around proteins to form chromosomes. And the chromosomes carry that genetic information inside the nucleus of eukaryotic cells. Now I want to go back to this base pairing idea. The order of bases in the DNA, or the DNA sequence, varies. That's what's different for each gene that makes a protein. It varies among individuals, it varies among species. Each species has some characteristic sequences in it. But if you know the sequence of one strand, you can figure out the sequence of the other strand because of the base pairing rules, or the Chargaff rules. They show you here one base pair. And so A always base pairs with T. That's the way they fit together. That's the way they form hydrogen bonds. And then C always pairs with G. That's the way they form hydrogen bonds. And so that's the way they pair up. So when a cell divides, when a cell makes 2 new daughter cells, when one big cell separates into 2 in that cell division or mitosis process, we have to replicate. Otherwise, in whatever species it was, it has 8 chromosomes in it. Fruit flies have 8 chromosomes. If they didn't replicate the DNA before they divided their cells, the next cell would have 4 and 4. And the next cell would have 2 and 2. And the next cell would have 1 and 1. And then what are you going to do? So in order for the daughter cells to be the same as the parent cells, the DNA has to replicate before the cell divides. So that we can take that one single sister chromatid and make a copy held together as a centromere, and those are going to pull apart in mitosis. When DNA replication occurs, each strand can serve as a template to synthesize a new strand. Because we have these base pairing rules, when we take a single strand, we know what should fit with it because of hydrogen bonds. So DNA replication is going to result in 2 double-stranded DNA molecules that are identical to the one that we with. Now there's some enzymes that help this process take place. We know they're enzymes because their names end in A-S-E. The first thing we have to do is unwind that double helix, and that uses DNA helicase. And then DNA polymerase polymerizes or forms each strand. It uses a template of the first strand to make a complementary strand. And we're going to look at how that happens in just a second. And then DNA ligase seals any of the gaps that are there. So this is a figure from your book that's showing this. And it starts off with the 2 strands of the DNA molecule. They're only showing you a portion. They're only showing you 4 bases of the sequence. To keep it simple, and also that's enough to show you how the process works and to show you how they match up. So the 2 strands of a DNA molecule are complementary. Nucleotides match up according to base pairing rules. C pairs with G, and T pairs with A. T pairs with A, C pairs with G. And so when we start to replicate the DNA, the 2 strands unwind or separate. The DNA helicase helps them. And they move apart slightly because there's an enzyme that's going to come down the middle and synthesize the new strand. That enzyme is the DNA polymerase. Each of these blue parent strands serves as a template to fill in these DNA, the new DNA strand. And it does that according to the base pairing rule, according to what can form hydrogen bonds. If it can't form hydrogen bonds, then it won't get linked in there. If it can form hydrogen bonds, then it'll get linked to this new growing strand. So wherever there's a G, a C will get added. Wherever there's a T, an A will get added. Wherever there's an A T will get added. Wherever there's a C, a G will get added. And then the DNA ligase fills the gap. And so what we end up with are 2 new strands. Now, if we look at what we started with, and then we look at these 2 DNA strands, they're all exactly the same. Now, in these 2 daughter strands, in these 2 daughter DNA molecules, one strand is the parent strand, and one strand is a new strand. And that's the case for both of them. And so we sometimes say that this is an example of semi-conservative replication, because we somewhat conserve the DNA. One strand is old, conserved. The other strand is new, newly synthesized. And so there's a couple of other figures that are showing you this exact same thing. So we had to unwind the DNA. Here we had to unwind the DNA to fill in these new strands. It's called the semi-conservative model of replication because it ends up with one strand being new and one strand being old. And the physical way that it happens is whichever 2 nucleotides can form hydrogen bonds with each other or base pair with each other, that's the nucleotide that gets added to the growing strand. So we're using the parent strand as a template to make a new strand of DNA. Now, sometimes mistakes happen. The incorrect nucleotide gets put in. So there are some DNA repair mechanisms that help fix DNA that's been made incorrectly. The DNA polymerase enzyme itself has a proofreading function. It can figure out if it doesn't match up correctly. It can figure out if the nucleotide that it just added doesn't have the right structure. The enzyme makes sure that the nucleotide that's being added matches the nucleotide in the template strand, and if it doesn't, it takes it out and it puts the right one in. And then there's some other DNA repair mechanisms. There are several processes that use enzymes to repair DNA damage. But sometimes we don't catch those errors. Sometimes that polymerase doesn't work or some of the other processes don't work, and so we don't correct the errors. So we get these uncorrected errors in the DNA replication, and those become sometimes what we call mutations. And a mutation is a permanent change in the DNA sequence. And we're going to cover mutations in a little bit more detail toward the end of this because they're covered in more detail in Chapter 7. But they can come from these mistakes that happen in DNA replication. And so that's one of the reasons why it's being discussed at this part in Chapter 6. So then we move on in Chapter 7 to look at some of the other things that DNA does. Chapter 6 talks about DNA structure and DNA replication and how DNA tries to be really accurate when it's replicated. That DNA polymerase enzyme tries to not make any errors. And then Chapter 7 is going to get into the other thing that we can do with DNA, which is use it to make proteins. DNA carries all the genetic information that we need to make all of the proteins that are in a new organism. And that genetic information consists of the sequences of the bases. Now, that sentence probably isn't going to make too much sense to you right now, but it probably will make sense to you after we go through this next segment, because it's the sequence of the bases that determine which amino acid gets put into the protein. So we have these long DNA molecules. They're made up of genes. A gene is a segment on the DNA molecule, and it specifies the structure of an RNA or a protein product, and that information that it carries is in the order of the nucleotide. You'll see in just a little bit why the order ends up being so important. We can talk about some terms. When we take a gene and we turn it into a protein, that's called gene expression. We have lots of genes that are not expressed. We have lots of genes that never become a protein. You have the same genetic material in all of your cells. Some cells turn that gene into a protein. Some cells turn different genes into proteins. So gene expression is the process by which the information in the gene, the order of the nucleotide, becomes converted into an RNA or a protein product. And that takes two steps. The first step is transcription, and the second step is translation. Transcription takes the information in the DNA and makes an RNA molecule or an mRNA molecule. And translation takes that mRNA and turns it into a protein. And so your book takes you through the steps of transcription and translation. The gene's nucleotide base sequence has the instructions for building the RNA and then the protein. We know that DNA is in the nucleus. From our study of organelles, we know that DNA is housed in the nucleus of the cell. We know that proteins are made on ribosomes that are in the cytosol, cytoplasm, or on rough ER. So if the information is in the nucleus, but the proteins are made on the ribosomes, how does the information get from the nucleus to the ribosome? And the answer is that the cell makes an RNA called an mRNA or a messenger RNA, and the cell makes that copy because it has the same information. We'll see more about what that means in just a minute. But the mRNA can leave the nucleus. And the mRNA leaves the nucleus and takes that protein building instruction to the ribosome. Now, this is one of those topics that we do a little piece at a time. We kind of try to layer on detail after detail to not be overwhelming. But sometimes some of the statements don't make sense until you go back at the end and you have the complete picture, and then you can go back and say, okay, that makes a little more sense. So we're taking the information in the DNA, we're making a messenger RNA molecule, and then we're making a protein. We don't have enough detail really to know what that means. But once you have some of that detail, some of those previous steps make a little more sense. So here your book introduces the structure of RNA. RNA is very similar to DNA. They're both nucleic acids. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid, which means the only difference in their structure comes right here. There's an oxygen here that's not where we get the deoxyribonucleic acid. Other than that, these structures of guanine are the exact same. Those little differences make the difference between DNA and RNA. And DNA is usually single-stranded, where RNA is double-stranded. So, hang on, I might not have said that. I might have said that backwards. RNA is single-stranded, whereas DNA is double-stranded. I hope I said that right to begin with. So RNA, because it's single-stranded, kind of breaks down a little bit easier, but we don't need it for very long, just as long as it takes to get a protein translated. Now, the RNA nucleotides follow very similar base pairing rules. The RNA nucleotides follow very similar Chargaff rules or base pairing rules to ones that the DNA has, with one exception. There is no RNA nucleotide that has a T on it. There is no T in RNA. Instead, it's replaced by a U. So in the DNA, for replication, wherever we had AT, TA, GC, in the DNA, wherever there's a T, we will put an A. That much doesn't change because there is an adenine nucleotide with the RNA sugar on it. The C and the G are exactly the same, and the G and the C are exactly the same. Where it becomes different is when we have an A in the DNA. If we were doing DNA replication and making a DNA copy, we would put a T. But there is no T in RNA. Instead, it's replaced by U. U for uracil. And so in the RNA, now there are no T's. You can look all the way through here and you don't see any T's. All the T's are replaced by U. Wherever you have an A in the parent strand, we put a U in the RNA. So the process of transcription is taking a DNA and making an RNA. Use an enzyme called RNA polymerase. It's going to work very similar to DNA polymerase. But where DNA polymerase was synthesizing a DNA molecule from a DNA template, RNA polymerase synthesizes an RNA molecule from the DNA template. The RNA polymerase binds at the beginning of the gene. It's the enzyme that carries out transcription. The sequence that it binds to is called the promoter. So the promoter is in the DNA, and it's the sequence where the RNA polymerase binds. And then the polymerase moves along the DNA It unwinds the DNA, and it fills in the RNA by linking those nucleotides in the order determined by the template strand. And the only difference from our previous example is that instead of T, it places U. So this new mRNA ends up being a copy of the gene. So there's a figure from your book that shows it. It starts out by showing you the DNA and the the RNA polymerase binding to the DNA. This figure, they show you across 2 pages in your book. They show you across page 118 and 119. And so they show you the gene sequence, and the promoter is right before the gene sequence, and that's where the RNA polymerase binds. It unwinds the DNA and fills in the RNA. Your book uses the same color convention, so it always shows DNA strands in blue and the RNA strands in brown. And so they show this RNA being developed from this DNA. If you look at it pretty closely, you can see the same base pairing rules we looked at on the previous one. Anywhere there's an A in the DNA strand, there becomes a U in the RNA strand. But all of the other base pairing is the same. Here's a big version of that slide that shows the DNA unwinding. And in this space, we put the enzyme and we start making an RNA, a messenger RNA. Then there's also a legend that tells you what's happening as you look at the figure. The polymerase begins to move along the DNA, it unwinds it, then it links RNA nucleotides into a strand of RNA. It does that by matching them by hydrogen bonds to the DNA ones, so it ends up in the same order. This is sometimes called a transcription bubble because it looks like it has this bubble, it looks like it has this space in it. And then the next figure is a scanning electron micrograph that's showing that there's many polymerases. So there's many enzymes that can transcribe a gene region at the same time. This thin line, this thin strand is the DNA molecule, and then each of these is an RNA transcript that's coming off of it at the location of an RNA polymerase that's polymerizing it. So because we have that base pairing, we can end up with an RNA transcript that has almost the exact same information that the original DNA did. So I'm going to go back to this slide to really help you understand that. If we had replicated the DNA, then this is what we would have gotten for our second strand. But if we do transcription, this RNA is what we get for that second strand. And if you compare those two, they're very similar. The only difference is wherever there's a T in the DNA, there's a U in the RNA. So when we made the statement that we take the information that's in the DNA and put it into RNA, that's what we meant. When we say we make a copy of the DNA that has the same information that was in the gene, the RNA has the same order of nucleotides that the DNA has. So transcription is just making an RNA copy of the DNA. The transcription piece is just making this RNA copy using RNA polymerase to do it. So now that that mRNA is formed, and it has that same information that was in the original gene, it can leave the nucleus. That nuclear membrane has pores, and that mRNA can go out those pores and leave the nucleus. And it can go to the ribosome, which is the site of protein synthesis. It can either go to free ribosomes in the cytoplasm or to a ribosome that's on rough ER. We can take that information that's in that RNA and turn it into a protein, and that's called translation because you're translating the message into a protein. Now, there's different types of RNA that are involved. The mRNA is the messenger that was produced by the transcription step. It has that protein building information because it has the same information as the DNA. The other two types are called ribosomal RNA. They help form the structure of the ribosome, and transfer RNA. So when we look at the mRNA to make a protein to do translation, we look at the nucleotides in sets of 3. And the 3 nucleotides are called a codon. So the information in mRNA is in sets of 3 nucleotides. And that's called a codon because it codes for an amino acid. So we have to go back and think about our macromolecules just briefly from chapter 2. A protein is a string of amino acids strung together to make a big long protein. They have primary folding and secondary folding and tertiary folding and quaternary folding, all those levels of protein folding. So if we're making a protein, we're stringing together those amino acids. That information is what's in that mRNA. Every 3 nucleotides codes for 1 amino acid, and we call it a codon. It's a nucleotide-based triplet that codes for an amino acid or a stop signal during translation.